This video will help you to understand the training of our Retina AI model. For the collection of the data, we have collaborated with the Center for Sight. From here we collect data in two forms. 1. Image of the register which contains the patient details and information regarding their medical conditions. 2. Images of retina scans which we get in a pen drive. Now in this step, we are going to manually enter the patient data from the collected image to the CSV_retinal_data Google Sheet, which contains information like patient name, MRD number, their sex, and age of the patient, and whether they have hypertension, cardiovascular disease or diabetes. After entering the data, we will download this Excel sheet as a CSV file and place this file in the Retina project directory. Now we are going to copy the data from the USB pen drive to the data set folder in Retina project directory. Now let's understand the Retina project directory. In Retina project we have the following folders. 1. CSVs folder which contains two folders, CAD and HTN_CSVS. These folders contain the CSV files we get after pre-processing of the data from the CAD and hypertension model respectively. 2. In dataset folder we have folders of all patient data which contains the retina scans of patients. 3. DL dataset folder contains train test data for deep learning. 4. Labeled_data folder contains the data we get after the labeling process. 5. The notebook folder contains all the notebooks required for the training of the model, and the archive folder contains the backup of the master notebook. 6. Octa 500 data folder set contains the extra data set. 7. Retina model training folder contains videos and files related to the model training video. 8. The saved model folder contains the model saved after the training of both CAD and hypertension models. 9. The sheet photos folder contains the sheet photos we get from the Center for Sight while data collection. 10. The tech doc folder contains all the technical documents of the project. 11. The training results folder contains the record of the training of both the CAD and hypertension models. 12. Finally, the retinal_data_preprocess.csv file contains the patient data and their medical conditions. Data preprocessing. Before starting running the code first, please make sure all the file paths given in all functions are correct. If these paths will not be correct, then it will throw us a file not found error. To start the data preprocessing, we are going to run the master notebook. First we will import the libraries, then we are going to load the user-defined functions, then run the crop_and_label function to start cropping and labeling of the retina images. This data preprocessing step will take about 1 hour to complete. At this point, cropping and labeling of the data are done. The cropped images will be saved in the dataset folder in the respective folder of the patients. And the labeled data will be saved in the labeled_data folder. Biomarker calculation. After labeling the data, our next task is to extract the biomarkers. In our literature study, we identified 4 biomarkers: area of the foveal avascular zone, FAZ, distortion of the FAZ, flow density, or perfusion density, and vessel length density. Changes in the values of these markers are indicative of cardiovascular diseases. Now we are going to calculate the biomarkers of the patients by running the code in biomarker extraction. First, we will calculate the biomarkers for the CAD patients, and then we will calculate the biomarkers for the normal patients, calculation of CAD biomarkers will take approximately 30 minutes and calculation of normal patient biomarkers will take about 4 to 5 hours. Now the biomarkers have been calculated. We have got two CSV files named CAD.CSV and normal.CSV in the CAD_CSV folder in the Retina project directory. Merging of CSV files. Now we're going to merge both the CSV files we got after the biomarker calculations because the CAD.CSV file contains the data corresponding to class 1 and the normal.CSV file contains data corresponding to class 0. Now to merge both the CSV files we need to run the code in CAD_CSV_prep_ML notebook. Please make sure to input the correct input file path for error-free execution of code. This code will merge both the files and give us the final patient data ML CSV file. Now that we have got the patient data in the CSV file which contains the biomarkers of the patients of both classes. Now we are ready to train the machine learning model of the CAD Retina project. We can also see the description of the data like scan quality of the images, number of patients with CAD, hypertension, and diabetes disease in the same notebook. To see the details of last 100 patients we can input the desired number in the plot_x function. This function will give us the bar graph of the last X patient data entered in the sheet. Now we are going to train our machine learning model. To train the ML model we are going to run the CAD_ML_Retina notebook. First we are going to load the final patient data ML CSV file, and for pre-processing we are going to remove the data where FAZ circularity is 0. Then we are going to check for the class imbalance in the dataset, as our class distribution ratio is 9:1. We will use oversampling techniques to balance our target class. Then we are going to use standardization, PCA, then we are going to train our machine learning models. This process will take about 5 minutes to train the ML models. The trained model will be saved in the CAD_models in the saved model folder. Now let's train our deep learning model. To train the deep learning model we will again go to the master notebook and run the code of deep learning dataset creation. This code will remove the red and green lines from the images, and split the dataset into training and testing images. This process will take about 1 to 1.30 hours to complete. Now the data has been splitted into training and testing dataset, we can start training the deep learning model. To train the deep learning model we are going to run code in the CAD_DL_Retina notebook. For deep learning, we have used PyTorch, which is an open source AI library based on the Torch library, used for applications such as computer vision, deep learning and natural language processing. First we will import all the libraries and then we will initialize the data augmentation process and do the image processing. After that we will create our deep learning model. For model creation we have used Inception v3 architecture. The model training process will take about 2.3 hours to complete. After following all the steps, we have trained both the machine learning and deep learning models of the CAD Retina project. To see the performance of the model we can have a look at the classification report for the machine learning model, and we can see the accuracy, precision, recall, and F1 score of the model. As we can see in the classification report, for Class 1, CAD, precision, recall, an F1 score are 0, which means that model is not being able to differentiate between CAD and normal patients, and it is predicting CAD patients as normal patients, which is incorrect. Model accuracy is 91% because the model is predicting the normal class correctly and the CAD class incorrectly. For example, if the data balance would have been 50 to 50% for both classes, CAD, normal, the accuracy would be close to 50% in case of the model not learning properly. For the performance of the deep learning model, we can see the training epochs, training, validation, and test accuracy of the model. We can also have a look at explainable AI. In deep learning, where researchers cannot explain why the model arrived at a particular decision, the explainable AI helps to understand, interpret and trust the predictions made by the AI models.